Okay, so we will start from the chapter 7. So do you have any other question in chapter 7 in course or the homework? Do you have any other question? Okay, so we will start our class right now. Okay, so chapter 7 is linkage and recombination and eukaryotic gene mapping. Okay, chapter 7 is composed of the 5 subjects. Okay, let me see some details. So previously we learned the principle of segregation, and also we learned principle of independent assessment. So based on the principle of independent assessment, we have the 9:3:3:1 ratio. So F2 progeny, the ratio will be 9:3:3:1 ratio is like this. Okay, so this is we have to know this concept. So usually the independent assessment, okay, so this is the parent generation, large AABB, and small AABB is also parent generation. Their gametes will be, this one will be large A, small b, small a, small b is like this. So after fertilization, we have the F1 generation, large A, small a, large b, small b is like this. Okay, so to make the gamete, okay, so for example, this is the one case. One case will be the large A, large b, or small a, small b. So this is the original combination of allele. So because this is exactly same as the large AB and exactly same as small AB. Okay, so this one we call it as original combination. However, this case, small a, large b, or large a, small b this is new combination. Previously, we don't have the large a, small b. Also, we don't have a small a, large b. So this is we call it as the new combination of allele. So this is, we call it as a recombinant gamete. Okay. So this is the linked gene. Okay. For example, large A, small b is located on the same chromosome. So in this case, okay, in this case, okay, so in this case, and also, in this case, we call it as a linked gene. So locate close together on the same chromosome. Okay, so if large A, small b is not linked, this is independently, they are located independent chromosome. So in this case, we call it as a not linked. But still the genotype is exactly same, large A, large B, small b. This one is large A, large B, small b is like this. Okay, but linked gene is they are located in the same chromosome. Okay. Okay. So if they are not linked, usually in F2 generation, we have the 9:3:3:1 ratio. Okay, so if they are linked, so complete linkage, they made a non-recombinant gamete and non-recombinant progeny. However, crossing over happens, so they made a recombinant gamete and recombinant progeny. We will see some detail in the later. Okay. Okay, so this is the previously we learned the linked gene in the males 1 and the males 2. Okay. So usually the crossing over happens during the meiosis 1, especially the prophase 1. Okay, so usually the crossing over happens. So it will make the crossing over lead to the recombination. Okay, so this is the test cross to show that the demonstrate whether two loci are normally linked or not. Okay, in this case, M gene and D gene is located at the same chromosome. They are linked to each other. Okay. So large M means that they have the normal leaves. Large D, they enclose TOR. So we did the test cross with the recessive one with mottled if they have this kind of the dot, okay, and also dwarf means that they are very small, okay. If they are in the complete linkage, okay, this one they will make the large M large D or small m small d. After fertilization, one will become the normal leaves and TOR. One becomes the mottled leaves and dwarf. Okay, so with complete linkage, only non-recombinant. This is non-recombinant. Okay, this is exactly same as parents. This one is also exactly same as the parents. Only non-recombinant progeny is produced. It's like this. Okay. Okay, so if they are unlinked, so M and D, if they are unlinked, okay, if they are unlinked after we did the test crosses, so we will have the 1:1:1:1 ratio. One will be the non-recombinant, another one will be non-recombinant, and the third one will be the recombinant. They have normal leaves, but they are very short. Another one is the recombinant. They have mottled leaves, but they are very tall. So in our homework, we cover this one. Okay, so number 15 is ask about, so if they are two genes is located in the different chromosome, their ratio will be 1:1:1:1:1, like this. Okay, very important. You have to know. You have to know. So if they are unlinked, they have the 1:1:1:1 ratio is like this. Okay, so if they are linked, okay, A and B gene, they are linked. So usually the crossing over happens. Okay. Okay, usually the crossover happens. Okay, so in this case, one will be the large A, large B. This is non-recombinant. Another one is a small a, small b. This is non-recombinant. But this one, large A, small b, small a, large b, will be the recombinant. Okay, so single crossover takes place between only two or prochromatids. Okay, so gamete received these two non-recombinant cases, and herpes two is recombinant cases. It's like this. Okay, and also we can calculate recombination frequency. Okay, based on the total number of progeny. Okay, so number of recombinant progeny is divided by total number of progeny multiplying 100. Okay, so in this case, large M, large D is they are linked to each other, and the genotype large M, large D, and small m, small d is like this. If there is no crossing over, they will have two gametes. Okay, one will be large M, large D. The other will be small m, small d is like this. But when crossing over happens, okay, when crossing over happens, okay, so this one, when crossing over happens, so we know that in this case, crossing over happens, two cases will be non-recombinant, two cases will be recombinant, okay? So it means that 50% will be non-recombinant and 50% will be recombinant, okay? Non-recombinant is large M, large D, small m, small d. Another recombinant case is large M, small d, small m, large d is like this. Okay, so based on this one, we can calculate the recombinance frequency, total number. So recombinant progeny is 8 and 7. So 8 plus 7 is divided by total number is like this. This is recombination frequency. So based on this one, so in this one, okay, this one is non-recombinant because the number is higher, and this one is also non-recombinant, number is higher, and this one is a number is small, this is a recombinant, and this is a recombinant. Based on number, we can calculate percentage of recombination, okay, 19 plus 16 divided by total number like this. 4% is the recombination, percent of recombination. Okay, so you need calculator, okay? You need a calculator, okay? 김건영, 지난번에 calculator도 안 가지고 뭐 시험 보는데, 근데 성적은 잘 나왔어. 오케이. 수업도 안 들었구나. Okay, like this, important, okay, you have to solve it, okay? Okay, another one is like this, okay. Now we will learn the allele in corporal configuration, in the allele in the repulsion configuration. Okay, so in our homework, we covered this one here. This one, number 28 is the ask whether this is coupling or whether this is repulsion. Okay, important, you have to know, important, okay? So what is the allele in coupling configuration? So we call it as cis configuration. So it means that one chromosome contains both wild type alleles. Okay, so in this case, and another chromosome contains both mutant allele. Okay, in this case, we call it as P plus, B plus. Another chromosome, they have a small p, small b is like this. So in this case, we call it as the cis configuration. To check whether they are cis or whether they are trans, we also can do the test crosses. Okay, we crosses with this small p, small b, and small p, small b is like this. Okay. Okay, so this one will make the non-recombinant, and they will make the recombinant is like this. Okay, except that the fertilization is happens. So one will become large, small p plus, b plus. Another one is small p, and small b is like this. Another case is like this. So, and also another case is like this, Okay, so so this is the non-recombinant progeny because their phenotype is exactly same as parents, and this phenotype is exactly same as parents. So this is important. If the phenotype of non-recombinant progeny is the same as the phenotype of the parents, we call it as a cis configuration. Okay. Another case is like this. Allele in the repulsion configuration. So wild type and mutant alleles are found on the same chromosome. Okay, so in this case, P plus B or small p plus B plus is like this. Their phenotype is they have the green and they have the brown or puparium is like this. Okay, exactly same as the phenotype is exactly same for their gene. This case is in their coupling configuration. In this case, they are in the repulsion configuration. So in this case, their non-recombinant will be like this, and recombinant will be like this. Okay, and they will cross with each other. Okay, so this one is that this one is non-recombinant progeny because their number is higher, 40 and 40, and this one is 10, 10 this is the recombinant because their number is small. But important thing is in this case, they are green thorax and brown puparium. They have P plus, B plus, and this one is purple thorax and black puparium. The important thing is the phenotype of recombinant progeny is as the same as phenotype of parents. In this case, we call it as repulsion configuration. Okay, so let me see our homework. So this is the progeny crosses large A, small a, large B, small b. So we don't know their configuration, but we did test crosses. Okay, so we have the 5 large A, large B, small b. Okay, so this is exactly the same as parents. Another one is this one is also exactly the same as parents. Okay, so it means that recombinant progeny is exactly the same as the parents. So in this case, we call it as a repulsion. We also can calculate the recombination frequency. Okay, total is 100, but the recombinant one is 5 plus 5, so it will be 10%. Okay. Okay. So this one is important. Okay. Okay. So we summarize it. If independent or assessment 1 to 1 to 1 to 1. Complete linkage 50-50. So linkage with crossing over. This case is more than 50%, and this case is less than 50%. Okay. Okay, so now we can predict the outcomes with the linked gene. Okay, so if we have the recombination number, we can calculate it is like this. Okay, now this one is very important. Okay, testing independent or assessment. Okay, we can use the chi-square test of independence. We can chi-square test of independence determines if two genes are sorting independently. Okay, so important in the final term exam, you have to solve this question. Okay, so in this cases, this is heterozygote, but their genotype is Y plus Y. and CB plus and CB is like this. Their phenotype is brown body and straight wing is like this. So they did test crosses with two recessive one, okay, but they have this number 63, 28, 33, and 77. Based on this number, we can do the chi-scale test of independence. Okay, previously we learned that if they are independently assorted, they will be 1, 2, 1, 2, 1, 2, 1. If they are comparative linkage, they will have 50, 50. If they are the linkage with the crossing over, they will have the like this. Okay, so we will test it. Okay, the important thing is that, okay, based on this number, we have to make contingency table. So construct table. So in this case, the y plus y, we can segregate it. So y plus and y, after this one, they only have the y, small y. So one case will be the y plus y. The other case is the y plus y is like this. So another case is we can also segregate this one, CB plus CB, and this one they only have a CB. Okay, we can make it. Okay. So okay, so we can make the graph. So in this case, 63, 33, 28, 77. So we can calculate it like this. Okay. Okay, after that, we can do the assumption. Our assumption is the segregation of the allele at one locus is independent. They are independently assorted. Based on this assumption, we can calculate the expected number. So this is expected number. So expected number total is row total multiply column total divided by grand total. Okay, so in this case, y plus y, CB plus CB. Okay, this one. So this is the observed number. So we can calculate the expected number. Okay, expected number is 96 multiply 91 divided by 201 is like this. Okay, we also can calculate the expected number is like this. Okay, after that we can calculate the chi-scale is like this. Okay, this is chi-scale number. And also, we can calculate the number of freedom. Number of freedom is the number of rows minus 1 multiplies number of column minus 1 is like this. In this case, df will be the 1. Okay, based on their df is 1, our number is, we calculated 30.73, their number is like this. So it means that probability is less than 0.005. Okay, so when the probability of chance is less than 0.05, chance is not responsible for difference. Okay, when the probability of chance is less than 0.05, chance is not responsible for difference. It means that they are not independently assorted. Okay. Okay, so it means that the gene for body color or the gene for type of wing is not assorted independently because the chance is not responsible for difference. Okay, it means that they must be linked. It's like this. Okay. Okay, so based on this one, so number 16 is exactly the same as the previous one. Okay. Okay. So in this answer, we can make it large A, small b, large b, small, because you can write down the BG and the SPS. It depends on you, okay? So first of all, we have to calculate the chi-square number, and also we calculate the number of freedom. So based on this one, the P is less than 0.05. So it means that we can reject independent or sourcement. It means that two genes they are linked to each other. Okay. Okay. You have to know, okay. And also based on the recombination frequency, we can measure their distance. Okay, you have to know. And also there are several questions asking about the recombination rate and also the order of gene on the chromosome. Okay. And also because there are two strands, the double crossover happens. In the case double crossover happens, we cannot detect it. So it means that it can underestimate the true map distance. So we cannot find these cases. Okay, so it means that underestimation of true map distance happens. Okay. Okay, so this is the 2-point test cross with between gene. So 2-point test crosses, we have the recombination frequency. If their recombination frequency is 50, it means that they are independently associated. It means that they are located in different chromosomes. Okay, so and also double crossover made on the estimation of true map distance. Okay. Okay, so C and D is the 28 was shown recombination frequency, but actual will be the D and B will be the 10, and B and C will be the 20. Actual will be the 30. It's like this. 28 comes from underestimation of true map distance. Okay. Okay, so based on this one, okay, we calculated their location is like this. Okay, you have to know. This one is also you have to know. This one is also you have to know. Okay. You have to define that their location is like this. Okay, 28 is asking about the repulsion and coupling. The final one is 29, this one. Final important thing is this one. Okay. So in this case, 3-point test crosses. Okay. We cover 3 genes, ST, scarlet gene, and the E gene, and the SS gene. Okay, ST is involved in the wild type color is like this, but ST they are very bright, and also E means that wild type their body color is brown. However, ebony E will be the black is like this. The bristle SS wild type is very long, but spineless they are very small, while they don't have the bristle is like this. Now we will the 3-point test crosses cross with the 3-1 is recessive is like this. Okay, based on this number, we can calculate their location, and also we can calculate their distance. This one is very important. Based on this one, we have the 1, 2, 3, 4, 5, 6, 7, 8 cases. Okay, usually we have the only 2 cases. This one, non-recombinant cases. First of all, you have to identify non-recombinant progeny, two most numerous phenotype. This one is the non-recombinant progeny. Important, you have to know. Another one is identify double crossover, two at least numerous phenotype. Before that, you have to know this one. So if double crossover happens, the only thing is that double crossover made only middle of gene is altered, is like this. When double crossover happens, this one is not changed and this one is not changed. Only the middle gene is changed, is like this. Okay, this concept is very important to solve this one. Okay, so identifies double crossover. The smallest one is like this, and compare each other. Okay, for example, this one wild type, they have this one, this one, this one. This one mutant, they have this one, this one, this one. So this one is that spineless. Okay, they are wild type, but this is their spineless. Okay, this one is they have scarlet and ebony. So this one is they are recessive. This is wild type. So when you compare this one and this one, the difference is the spineless. When you compare this one, okay, their difference is spineless. It means that spineless gene is located in the center. Very important. So SS gene, based on this number, we can find we can say that SS is located in the center. At that, so we can determine the location of crossover. At that, so in this case, they are single crossover, and in this case, they are double crossover is like this. Okay, so this symbol indicates the position of crossover is like this. Okay, at that we can calculate the recombination frequency is like this. So the ST-SS recombination frequency. Okay, so ST and SS recombination frequency. Okay. So total is this one is ST-SS. Okay, so ST-SS. It means that 50, 52, 5, and 3. How about the SSE? SSE is this one. The recombination frequency is like this. So based on this number, we can say that SS is located here, ST is located here, E is located here, but distance is 14.6. Distance will be the 12.2 is like this. Okay. You have to understand this one, okay? So this one is also asking about exactly same as this one. So you have to find the recombinant, non-recombinant, and double crossover. Okay, and you have to calculate the distance like this. Okay, and also important thing is this one. You also have to calculate the coefficients of coincidence. So coefficient of coincidence means that a number of observed double crossover is divided by number of expected double crossover. Okay, number of observed double crossover is the 5 plus 3, and number of expected double crossover is the total number, and frequency of the frequency of the recombination ST-SS recombination frequency 14.6. So it will be the 0.146. Another will be this way. This frequency is 12.2. So this one will be 0.122 is like this. So their coefficient or coincidence will be 0.6 is like this. So based on this number, we can calculate the interference. So the degree to which one crossover interfere with additional crossover. This is, we call it the interference. So divide 1 minus coefficient of coincidence is 0.4 is like this. Okay. So this is this one. This question is asking about the location of gene and the distance of this gene, and also we have to calculate it coefficient of coincidence and interference. Very important. So do you have any other question in chapter 7? Any other question? Just you need a calculator, okay? Okay. 전기공학과 최민준, 이현지.
네.
수업 잘 듣고 있어? 잘 이해하고 있어?
네.
듣고 있습니다.
성적 잘 나왔던데. 오케이. 윤홍빈, 장준원 잘 듣고 있어?
예.
오케이. 장준원은
네, 잘 듣고 있습니다.
이해가 돼?
네.
김나연.
네,
교수님. 이해가 돼? 아니면 좀 어려워?
이해됩니다.
그러니까 홈워크를 풀면서 이제 다시 한 번 더 봐봐, 알겠지?
네.
오케이.
네.
로니카? Do you have any other question?
Actually, I do have one.
Okay, so go ahead.
For the chi-square, I don't seem to understand. Like I'm able to do every like the steps, but the last thing the probability thing, I don't know what is like P.
Probability. This one. Okay.
On slide 21.
Okay, slide 20. Okay, slide 20. Okay.
Like what is 30.73 used for? This I'm not able to understand the left side.
Okay, so because previously we learned, so I did not explain it. Okay. So previously we explained the chi-square number. Okay. So in our before midterm exam, so I explained how to calculate this one, and is this one exactly same as before? So we have this number 30.73 is like this, and also this is number of freedom will be the 1, so they are located here. So we have to find where 30.73 is located. Because this one is located here. No. They are located here.
Yeah.
Based on this one, we can say that the P is the less than 0.005.
Okay, I understood now.
Okay.
Thank you very much.
Okay. You have to know. Okay, you have to know to solve the final term exam. Okay.
Yeah. Thank you.
Okay. Now we will go to the chapter 8. So, do you have any other question in chapter 8 course or the homework? Okay, so we will see some details. So chapter 8 is chromosome variation. So we have the 4 subjects starting from the chromosome mutation, rearrangement, aneuploidy, and polyploidy. And also we will learn some chromosome rearrangement, and we will cover aneuploidy and polyploidy is like this. Okay, so this is our chromosome. Okay. And we cover types of chromosomal rearrangement: duplication, deletion, inversion, translocation. So we have chapter 8 homework. So we have the question 20 is we asking about whether they are duplicated or they are inverted, whether they are you have to know whether they are deleted. Is like this. Okay, because this is a genetics class, so this is important in genetics class. Okay, okay, so you have to know. Okay, okay. So, and also when duplication happens, so they will make this kind of structure. And also deletion happens, they will make this kind of loop. Okay. And also when paracentric inversion happens, so they will make, what is paracentric inversion? Paracentric inversion do not include the centromere, but pericentric inversion include the centromere in inversion. Okay, so based on this one, you have I asked the case number 25. Okay, so initially this is wild type chromosome A, B, C, D, E, F, G, H, I. So okay, in this case, A is they have A, B, C, D, E, F again, and G, H, I. Okay, so based on this one, so in the pro page 1, especially in the meiosis, so you have to write down, draw the picture like this. Okay, you have to know when duplication is happen, so they will make this kind of structure. When deletion is happened, so in this case, B is they are deleted. So in this case, also you have to write down is like this. So this one is the paracentric, and this one is the pericentric. Okay, paracentric do not include the centromere. Pericentric is include centromere. Okay, so in this case, they are A, B, D. So A, B. So like this. Okay. So we can write down like this. E, D, C is located here. Okay. So you have to draw. You have to draw this picture. Okay. Okay. So because the D is already complicated, they are pericentric. Okay. So you have to know. You have to draw the picture is like this. Okay. Very important. Okay. So do you have any other question? Okay, so this one is important, and also this one is also very important because we are genetics class. Our chromosome structure is most important in the genetics class. Okay. Okay. Okay, and also we cover the haploinsufficiency, means that condition arise when the normal phenotype require the both proteins. Okay, so usually they require both proteins to make the smooth, but in this case they only have the one. Okay, haploinsufficiency, they make this kind of structure, and also haplosufficiency, they will make the obese mice is like this. Okay. So this is the paracentric inversion. Okay, so paracentric inversion is happens. They do not include the centromere. Okay. Okay, so they will make this kind of structure. So this kind of structure. After that, this one is the breaks is happens. So they will make this one. They have dicentric chromatid. They have the two chromosome, one chromosome, another chromosome. This one is, we call it a dicentric chromatid. So paracentric inversion, they made dicentric chromatids. After that, they will have the gamete. Two is non-recombinant, two is recombinant, but this is non-viable. It's like this. How about the pericentric? Okay, so pericentric inversion is happens. So they include the centromere is like this. Okay, so in this case, they will make the two. They make two structures like this. Okay, finally, they will make the two is non-recombinant, another one is recombinant, but they are non-viable. Okay, because they are non-viable because some of gene is missing or some of gene is too many copies. Okay. Okay. So dicentric chromosome is produced when crossing over takes place. In what kind of rearrangement? Dicentric is paracentric inversion. So they have the two centromere is like this. And also we cover chromosomal translocation, non-reciprocal and reciprocal or Robertsonian translocation is like this. Okay, and also this one is very important, translocation in the meiosis. Okay, important. Okay, so translocation happens between this is normal, this is another normal, but translocation happens. So one made the T1, they are composed of A, B, C, D, Q, R, S, T. Another one is the M, N, O, P, E, F, G is like this. Okay. So in propage 1, so they will make the cross-like configuration is like this. Propage 1, so A, B, C, D, G, F, E, in this case P, Q, and M, Q, R, S, T, right? They will make the cross-like configuration like this. Okay, so one case is they are segregated. In the anaphase 2, they will be segregated. So this one is alternative segregation, usually happen. So these two is segregated. So these two is segregated and these two is segregated together. This is alternative segregation, usually happens. Another one is adjacent. So they are, the upper one is go to the one cell and the other one go to the one cell. So in this case, we call it as adjacent to segregation. So this one will one cell, this one will go to another cell is like this. Okay. So based on this one, you have to know. So we have the question, this one. Okay, so crossing translocation happens between the CD usually they have CD, EFC, but they have CD, V, WX. In this case, they have TU, VWX, but they have TU, EFC is like this. Okay, based on this one, you have to write down this figure. This is for page 1, they are showing like this. Okay, you have to know, and also, so you have to diagram the alternate and adjacent 1 and adjacent 2 is like this. Okay, and also you have to write down their genotype is like this. Okay, because we are genetics class, our chromosome is very important and translocation is very important. Okay, so usually during the anapase 1, they will make cross-like structure. Okay, you have to write down, you have to draw the picture like this, very important. Okay, in the final term exam, you have to draw it like this. So one case will be alternate segregation. It usually happens. This one and this one go to the one cell, and this one go to another cell. And their genotype is the one will be the A, B, C, D, E, F, G. The other one will be the N, M, O, P is like this. Okay, another one, this case will be the A, B, C, D, Q, R, S, T. M, N, O, P, E, F, G is like this. Okay, so this is the genotype is like this. This one is their viable. However, adjacent 1, adjacent 2, they are very rare. Okay, most of them they are non-viable because some of them they missed some gene, or some of gene is the duplicating. Okay, important. Okay, important. Okay, Lonika is very important. Okay, very important. Okay. And also Fuji, very important. Okay.
네.
Okay. Now we will go to the aneuploidy or polyploidy. Okay. So aneuploidy and polyploidy, the cause, first of all, we will see the cause of aneuploidy. So non-disjunction is happens during meiosis 1 or meiosis 2 or during the mitosis. Okay, this is mostly happens. Some cases centromere is deleted, so they will make this one. Another case is a Robertsonian translocation. Okay, so you have to know the type of the aneuploidy because we are the genetics class. This is important to us. So nullisomy, so this is the normal cases. They are composed of 1 chromosome, 2 chromosomes, 3 chromosomes, and 4 chromosomes. There are 4 pairs of chromosomes. They are 2N. They are diploid. 1, 2, 1, 2, 1, 2, 1, 2. So nullisomy means that 2N minus 2. They don't have this one. Trisomy means that they have additional one, 2M plus 1. Monosomy, they only have 1, 2M minus 1. Tetrasomy is 2M plus 2, it's like this. You have to know, in our homework, we cover this one. So because they are 2N is 16. Okay, so when they are the trisomic, when they are double monosomic, when they are nullisomic, okay, you have to, when they are tetrasomic, you have to calculate their number. Okay, but important thing is you have to know this one first, memorize it. Okay, nullisomy is -2, trisomy is +1, monosomy is -1, tetrasomy is +2. Is it okay? Okay, so So in this case, 2n is 36, and trisomic means that 2n plus 1 means that number is 37 is like this. Okay. And there are many cases of aneuploidy, the capture of Zimson weed. So every case is aneuploidy, but they have different shape is like this. And also important thing is the trisomy 21. We call it as the Down syndrome. Okay, their chromosome 21 is they have 1, 2, 3 is like this. Okay, and also trisomy 18, 13, or trisomy 8, they have several cases is like this. And also uniparental disomies happen. Okay, because this is aneuploidy. Okay, sometimes in the normal cases they have this one, but this one they have this chromosome comes from both one parent. So this one we call it as a uniparental disomy. Okay, so in this case, mother is only one recessive, but their parents, their offspring will have this disease is like this. Okay. So, and also non-disjunction happens in mitotic division. So it will make this kind of patch. Some of the cell they have aneuploidy. Some of the cell is not aneuploidy, is like this. Okay. So non-disjunction in mitotic division, they generate patch of cell in which every cell has chromosome abnormality, while other patches in every cell has normal karyotype is like this. Okay. Okay, now we will cover autopolyploidy. You have to know autopolyploidy. Autopolyploidy means the presence of more than 2 sets of chromosomes. So autopolyploidies happen from one single species. So through non-disjunction in mitosis or non-disjunction is meiosis. This is 2n. Initially, their status 2n. So if no mitosis in the non-disjunction happens in the mitosis, so they will have the post-set of chromosome 1, 2, 3, 4. So this is, we call it as autotetraploidy 4n. Usually we have diploid 2n. No cell division happens, they made tetraploid. Okay. Another one is the non-disjunction happen in the meiosis, so they will make the triploid. 3n is like this. Okay, you have to know their terminology to solve the. So we know that autotetraploid. Okay, autotriploid, the n is 8, autotriploid means that 3n means that 24 is like this. This is very important in our genetics class. Okay. Okay. And another thing is allopolyploidy. Allopolyploidy. Presence of more than 2 sets of chromosomes, but from 2 species. So allopolyploid arises from hybridization between 2 species. following by chromosome doubling. Okay. Okay, so two spaces. One species they have 2n is 3, 6. Another species there 2n is 6 is like this. So they will make the hybrid is like this. So usually their gamete is ABC, GHI, but they hybridize it, and they will have the ABC, GHI. But usually the non-disjunction is happens. Okay, at that chromosome doubling is also happens. So their status will be the AA, BB, CC, GG, HH, II. Okay, their total number of chromosome is 12. So 12. So we call it as a 4n is 12. Okay, and this is the gamete is like this. Okay, so in this case, 4n is 12. We call it as an allotetraploid. Allotetraploid. Okay, tetra, 4. Okay, 4n is 2, 12 is like this. Okay, 2m plus 26 and 26, so they made the allotetraploid. 4n is 12. Okay, hybridization is happened between the two species. Have that non-disjunction is happens, have that chromosome doubling is happens. So they make the 4n is 12 is like this. Okay, so this is we call it as allopolyploidy. Okay. Okay. So this is the this is allotriploid. Okay, it means that usually the species A and species B, their n is 8, n is 7. Usually A, B, they will make 2n is 15 is like this. But this is allotriploid. So one case will be the ABB, the other case will be the ABA. So in this case, there will be the 22, and this case 23 is like this. Okay. Okay, so you have to know. So, for example, allotriploid, including species 1 and 2, you can calculate it like this. Also, autotetraploid in species 2, 428 is like this. Okay, you have to know. Okay, because we are genetics class, and this is very important to us. Okay, so chapter 8 is a little bit simple. Okay, you have to know this one. Okay, so you have to draw this kind. When translocation is happens, you have to know how to draw the picture is like this. And also when the pericentric inversion happens, their structure is like this. When paracentric is happen, is like this. When deletion, duplication, okay, you have to know, and you have to know what happens. So duplication is happen, deletion is happen, is like this. Okay, so do you have any other question in chapter 8? Okay, but important thing, you still have a chance to get A+, okay, if you get a good score in the final term exam. Okay, our midterm exam is very difficult to understand, but our final term exam, only chapter 7 is difficult to understand, but the chapter 8, chapter 9, chapter 10, chapter 11 is a little bit easy. So it means that higher chance to get higher point in the final term exam. Okay. Okay, do you have any other question? Okay, so today we finish class, and we will have another class and next Monday. Okay, so okay, bye.
Thank you. Bye.
Bye.
수고하셨습니다.
Okay.
고맙습니다.